Thank you, Miguel, for the opportunity to present this material. This is really kind of the goal of this is to try to explain to people how we investigate diseases of wildlife, including corals. And I think by getting this understanding, it might help you understand where we're coming from when we look at these issues. Just to give you a brief background, I'm a veterinarian and I run a field station for a laboratory that's based in Madison, Wisconsin, called the National Wildlife Health Center. And we're the only field station of the National Wildlife Health Center. We're located in Honolulu, and we do a bit of technical assistance, research, and the main goal of our existence is to help state and federal agencies address wildlife health issues in the Pacific Islands. So we're based in Honolulu, but we work throughout the entire Pacific. This map kind of shows you the area that we cover, which is pretty large. Why look at wildlife health and wildlife mortality events? Well, the major thing is that when you look at ecosystems, ecosystems tend to be resilient, and you'll have basically like a healthy coral reef, and it'll have a hurricane, and if it's a healthy coral reef, that coral reef will bounce back. But if you put enough insults on an ecosystem, eventually these ecosystems will transition from basically healthy to degraded in a rather rapid manner. And the Caribbean, the Western Caribbean is a really good example of that. In the early 1980s, you could go to coral reefs in Florida and they'd be covered with Acropora palmata and Acropora cervicornis. And because of a variety of ecological pressures, mainly human use, overfishing, land-based pollution, there was a whole series of coral reef mortalities that occurred, and they've lost about 80% of their croppers now. And those reefs have really not come back. So these are basically known as regime shifts, where they transition from healthy to degraded. And wildlife mortality events are signals that, hey, something's coming down the pike and you need to be paying attention. And if you don't pay attention to these signals, then you get these transitions to degraded ecosystems, and it becomes really hard to get them back to their original state. So this whole concept of the difficulty of getting an ecosystem back to its original state is called hysteresis. It's that resistance. So the way we come into it is we look at wildlife mortality events, and we're not really selective as to the animals we look at. So you'll talk to some people and they'll say, well, fish are really good ecological indicators of ecosystem health. And if you talk to people who look at marine mammals, they'll go, hey, well, marine mammals are really, really good ecological indicators. If you look at people who look at birds, they go, oh no, seabirds and land birds are really the good ones. We don't really care what animal is affected. What we care about is the mortality. And the tools I'm going to discuss here are really useful for a whole variety of different animal types. There are challenges to looking at wildlife mortality events. And one of these is that when there's a big mortality event, there's a lot of public interest and maybe resources go into looking at that event. But then that event will eventually wane over time, but the cause of that event is still there in the ecosystem and never goes away. That's something to keep in mind. And then the other thing is that we have a deficit of knowledge and detectability with size of animals. So, for example, mortalities of whales coming up on the beach, or sea turtles or seabirds, that's very visible. People can relate to those animals, and so those are likely to get reported. But things like fish and invertebrates, not so likely. Oftentimes, those mortalities won't get reported at all, which is unfortunate because when you think about ecosystem integrity, it's the lower trophic level organisms that drive the system, right? So we could lose whales tomorrow, but we still have the habitat. We lose coral reefs, we lose the habitat completely. So that's one challenge. The other issue is our level of knowledge also declines the further down you go in the phylogenetic tree. So we know a lot about marine mammal medicine, marine mammal physiology, marine mammal pathology. We know quite a bit about birds. We know quite a bit about sea turtles. fish and invertebrates, well, the knowledge base is not quite there, and so that's kind of a challenge as well. And when you look at it at the organismal level, when you look at this, when we're looking at wildlife diseases and wildlife health, we do a lot of comparative analogies. So, for example, you look at a whale. A whale has got heart, lungs, liver, just like a cow or a dog. So we can kind of relate what's happening in a whale to what we know about cows and dogs and horses. But when you start going down to the tissue level, things start breaking it apart. So a coral cell looks a lot like a dog cell, but a coral organism is really different than a dog or even a fish. So these analogies that we use to try to understand disease processes start breaking down when you're dealing with less conventional organisms. And when you get to the organismal level or the ecosystem level, those analogies break down completely. And so again, that makes it really difficult to understand processes in less characterized animals like corals. So when you have a mortality event, again, it's that signal, right? There's a decline in animal populations, and the fundamental question is, why are these animals declining? So you talk to biologists and you go, hey, how are your populations doing? And the biologists will probably say, well, I've gone out and done transects on the beach, and the nesting numbers are really good for these sea turtles, and so I think the populations are doing well. It's that when populations are declining, that people tend to overlook the importance of looking at the animal and the potential cause of death as an important factor. So, when you talk to biologists and go, yeah, the populations are going down and we're documenting they're declining, you ask them, well, why would that be? Well, these mortalities are natural processes and we can't do anything about them. And my argument is, you can do something about these mortalities, but you need to understand what's causing them. And there's a process that is useful that can be employed to understand the cause of these mortalities. And this is what I'm going to try to cover in this talk. So when you're looking at a mortality event in wildlife, when wildlife populations are declining, they're declining either because there's no reproduction or there's excessive mortality. And so there's got to be a reason why that's happening. And so the mortality is really caused by three things. It's an interaction between the animal that's being affected by the mortality, the causative agent, and the environment. And you have to have these three things interacting in order for mortality to occur. Okay? And so how do you get to the bottom of this? Well, understanding wildlife mortalities is a little bit like a detective investigation. So you think about, you have somebody who's dead on a desk with a knife in his chest. Well, what's going to happen? Well, first thing is you're going to get witness testimonies. Did somebody know this person? Did this person have any enemies? You're going to call the cops. The cops are going to come in. The detectives are going to come in. They're going to take fingerprints. They're going to take samples, send them off to the lab, get clues, and eventually they're going to figure out how this person ended up with a desk with a knife in his chest. Okay, the process in wildlife is kind of analogous. So in this case, though, we don't have the luxury of talking to the animals or getting witness testimony to figure out what's going on. So, for example, we have a dead dolphin on a beach, and people are looking around with their hands in their pocket, looking at the dolphin, and they're saying, well, what happened here? Well, you can't slap on a snorkel and go out in the water and interview other dolphins and say, hey, did you know this dolphin? Did he have a history of drinking or alcoholism or something like that? In veterinary medicine, that doesn't work. So we really need to depend a lot on field observations and observations that we get from collecting samples from the animal. And because there's so many animals that could be affected by mortalities, there's no way that I, as a veterinarian, can possibly know the biology and the ecology of all these animals and how they work. And I need to get a certain understanding of that in order to understand the process. And so this is why these disease investigation events are really best done by a partnership between biologists who know the animals, who know the ecosystem, who know the behaviors, who know the habitat, and then wildlife disease people who know pathology, medicine, biomedical information. And by bringing these two disciplines together, that's really the only way you can start getting some traction on understanding causes of wildlife mortality. So basically, there's two types of clues we look at in wildlife mortality events: field clues and clues from the animal. So 2 phases of wildlife disease investigations are field phases and laboratory phases, and I'll go through both of these to give you an idea of the questions that we ask when we look at this. So field observations are really, really important. Okay, so first of all, how do you know that there's excessive animal mortality? Are you monitoring that? You'd be surprised. Many people don't even monitor where and when animals are dying, but just getting that information alone can be extremely valuable over the longer term to try to determine whether there's a true signal there or not. So a good example of that was, back in Florida, in the early 90s, we were looking at a manatee mortality and trying to figure out what was killing manatees. And some of the resource agencies didn't even really have a good idea of what constituted an abnormal mortality events in manatees, because they really weren't taking good numbers of manatee deaths in the area. So, just simply enumerating when and where animals are dying, even if you don't do laboratory diagnostics, can be extremely valuable over the long term. And then you want to also describe the event in terms of place, and time. So animals, numbers of animals dying, are they abnormal or not? Species of animals dying, is it one species? Is it multiple species? That can give a clue as to what might be going on. So if only one species of animal is dying, you might be thinking, well, geez, maybe it's an infectious agent very specific to that species of animal. And then age and sex. Certain types of mortalities affect adults more than immatures. Certain types of mortalities affect females more than males. So you can think of the process that you've got this mortality event, you've got this super long list of all the potential causes of death that could be killing these animals. And my job is to take that list from 1,000 entries to hopefully one entry, which is the cause. And so I look at these clues to systematically eliminate possibilities based on what I see in the field and in the lab. Okay. Clinical signs are really super important. So on the left here, this is a photograph of a pufferfish that's puffed up on the surface. Normally, pufferfish don't do that. They don't puff up and float to the surface. If they're doing that, something is clearly wrong with that animal. So that's a clue. On the upper right, we have a duck here that can't hold its neck up normally. Clearly, this animal has got a neurological problem. So if I saw an animal like that, I'd be thinking, well, what are the causes of illnesses that could cause neurological problems in this bird? And I'd be maybe focusing on those. On the lower left here, we have a turtle with a big tumor on its head. So clearly that's kind of a problem, and I'd be thinking of, well, what could be causing tumors in reptiles? And then on the lower right here, we have an acropora coral in the Pacific with tissue loss. So you can see that the normal tissue is brown here, and the white area is areas where the tissue is left off the corals. Okay, so clinical signs are really valuable and can provide some clues. And then the place is really important as well. So how widespread is the mortality? Is it really localized to one area? Is it going over a huge geographic area? That can be important. And then any particular characteristics about the habitat that might have changed. So if there's a mortality event, clearly something has changed in the habitat. And what could that be? the phenology of the habitat. Maybe the plants have changed. There's new invasive species that have come in that the animals are eating. Maybe the way the animals are using the habitat has changed. All these are potential clues that could yield some information as to what might be going on. So for example, here we have a lake with a bunch of dead birds, and there's one lake on the left where the birds are scattered throughout, and there's another lake on the right where the birds are all clustered in one area. Well, what could be explaining this clustered pattern? Maybe there was a toxin that was spilled in this area and the birds got exposed to the toxin and they died. Maybe an alternative explanation is that the carcasses were all blown by the wind to the right side of the lake, okay? So these are all things that I, as a veterinarian, there's no way I can tell that. The biologist is the one who's going to be able to look at that and make those assessments. And so this is why it's really important to be aware of the habitat, how the animals are using it. Are the carcasses all decomposed or are they all fresh? That can tell you how long the mortality has been occurring. So anything, any particular characteristics of the habitat, land use patterns, change in land use patterns, change in the way people have been using the land, all those change in weather patterns, all those can be valuable clues that might help explain the mortality event. And then time is really important as well. So again, documenting animal mortalities over time can help you get an appreciation for what might be normal versus abnormal. So a good example, in Hawaii, we have seabirds, and seabirds are very synchronous in their life history. So for example, albatross will come in and they'll lay their eggs in September and October in the habitat. So we know that nesting season is September and October in the habitat. And so we also know that in January or in June and July, when the chicks fledge, there's a lot of mortality that occurs because we know that that's part of the life history of these animals. A certain number of these animals just don't make it because they starve to death, either because the parents abandon them or because the parents are inexperienced and are not able to feed the chicks adequately. So we know that every year we have chicks that die at a certain time of the year. If we have chicks dying at a different time of the year, then we know there might be a problem. That's not part of the normal life history of the animal. So again, having an appreciation for what's normal background mortality versus unusual mortalities is really, really important to help detection of these events. And it's important too, because if you can anticipate when mortalities are going to occur in a habitat, then you're prepared to collect samples. Because one of the big challenges in wildlife disease diagnostics is getting samples that are suitable for laboratory examination. I'll talk about that a little later. So, what kills wildlife? Well, same thing that kills a human being. It can either be an infectious agent or a non-infectious agent. And one of the big things that I found with coral reefs is that everybody assumes that corals are killed by infectious agents, but that's not necessarily the case. There's many non-infectious things that can kill animals. Trauma is a really good case in point. Trauma is one of the most important causes of death, both in wildlife and humans, I might add. Toxins and toxicants can kill animals, and those are non-infectious as well. And then corals can also be colonized by all sorts of different bugs. Microalgae, sponges can invade corals. So it's not all infectious agents, and it's really critical to recognize that. So how do we go about investigating wildlife diseases? This is probably one of the most important diagrams I'll show in this talk. We have a very systematic process that is applicable to both marine and terrestrial wildlife. And it typically starts with field detection. So biologist calls me up and says, hey, we have an unusual event. We have animals dying. What can you do to help us out? So I'll quiz that biologist about all those field indicators that I talked about earlier. But ultimately, you're never going to arrive at the cause of death of an animal unless you actually examine that animal itself. So you can do all the water sampling in the world. You can do all the sediment sampling. You can do all the sampling for the contaminants. All of that is going to yield useless information unless you actually look at the animal itself. And the process typically this starts out with doing a necropsy, looking at the animal, looking at the internal organs, and trying to figure out what in the animal is abnormal in those internal organs. That process is called pathology. So pathology is dissecting an animal, looking at the internal organs, looking what's abnormal, describing, abnormal shape, abnormal size, abnormal texture of the organs. And then critically, what we do is we take tissues from those organs, we put them on a microscope slide, and we look at those tissues at the cellular level. And that's called histopathology. And that process, pathology, will typically lead to a bifurcation. And will tell you whether you're dealing with something infectious or non-infectious. At that point, you can then bring other tools to bear to try to confirm the identity of the potential cause of death. So if you think you have a bacterial, for example, if you look at the slides under a microscope and you see bacteria associated with cell death in the tissues, then obviously you would look for bacteria and do additional assays to try to confirm the presence of those bacteria. You try cultures, maybe you do PCR to identify the bacterium. If you have something that's totally new, you might want to even do manipulations where you take animals in captivity and you expose them to the agent that you think is killing the animals. But the whole goal of these laboratory manipulations and captive studies, trying to reproduce the disease in captivity, is to reproduce the lesions that you're seeing in the wild, both at the gross and the microscopic level. So even these laboratory manipulations where you're exposing animals and trying to reproduce the disease in captivity, all of that is guided by pathology because you're trying to reproduce that lesion that you're seeing in the wild. Then ultimately, doing this process, you arrive at the cause of death, and then you develop assays to trace that cause of death, and then you start asking questions about how is that cause occurring in the environment? How is this thing being transmitted? How does it affect the habitat over time? So it's really critical to do this pathology because that's really, everything hinges on that pathology. And so when you look at any animal disease, I don't care if it's AIDS, tuberculosis, stony coral tissue loss disease, botulism in birds, everything hinges on pathology because the pathology establishes a definition of that disease. So you look at any animal disease, it has a case definition, and that you can look at that case definition almost like building a house. And when you build a house, you have a foundation. And if you have a weak foundation, that house isn't going to stand up. And that foundation is all based on the morphology of that disease. So that's why histopathology is so useful, because you can detect, almost always in pathology, you can detect infectious agents like fungi, bacteria, parasites. Those are very visible in light microscopy. So if those things are killing the animal, you'll see them on light microscopy. And then sometimes you can even detect things like viruses. Some viruses will make like little factories, and you'll see them on light microscopy. And if you can't see something on light microscopy, you can at least see how the host is responding to that insult at the tissue level. And so this is why it's such a valuable tool, because even if you don't see the cause, you'll see how the host is responding to that insult, and you'll see what tissue compartments are affected, and that can yield some very valuable clues. So I can't emphasize enough how useful and important pathology is to the whole process of wildlife disease investigations. So this is how pathology works. Basically, you have an animal, you cut it open, you look at the tissues under the grossly. So you can see here on this heart, there's a big white mass here that's clearly abnormal. We take tissues in formalin, in fixative, and then we look at them in a microscope, and then we characterize the process at the cellular level. So this gives you an idea of host response and potential etiologic agents, and gives you the critical bifurcation as to whether you might be dealing with an infectious or a non-infectious process. So it's kind of, it's field observations, characterizing morphology, arriving at potential causes. If not, then you keep cranking the crank So you have all these potential causes of death. You turn the crank. You may not arrive at the actual cause of death at the first cycle, but if you keep cranking that paradigm over time, you will arrive at the cause of death. So it may not happen initially, but over time, this process will work. There's other tools that we use. And so, for example, if we think we have bacteria involved in the mortality, then we can take tissue samples, and we can take those tissue samples and inoculate different media in attempts to grow a bacterium or a fungus and identify that fungus. So that can be very useful if you suspect that a bacterium or a fungus is a cause of death. You can also use PCR to identify these organisms. If you think you have a virus that might be involved, well, there's ways to grow viruses in the laboratory as well. Viruses are very different than bacteria and fungi in that they need living cells to grow. What they do is they go into the cell and they hijack the machinery of the cell to make more copies of themselves. So because of that process, getting viruses to grow in the laboratory is a lot more complicated than getting bacteria and fungi to grow in the laboratory, because in order for viruses to grow, you have to actually grow animal tissues on petri dishes. So you take animal cells, you grow these animal cells in petri dishes, and then you can grow viruses in these animal cells. Oftentimes this will work, but many times this won't. So this is a high stakes, high expense effort. You don't do this routinely unless you really have good reason to suspect that a virus might be causing the mortality of the animal. Toxicology, that's a process that we use if we think that a poison is responsible for the death of the animal. So in pathology, typically what you'll see for toxins is either you'll see no lesions at all, if it's an acute toxin, or you'll see particular patterns of cell death or organelle death in the cells. So if you suspect a toxin is responsible, you can extract tissues using different solvents or different reagents, and then you can analyze these extracts using very expensive machinery to find the toxin. So oftentimes people will call me and they'll say, hey, we have an animal dying, I want you to analyze for poisons. Well, my answer will be, how much money do you have? Because doing toxicology can be really, really expensive. So typically you don't do toxicology unless you have a really good suspicion that there's a toxin that the animal could be exposed to. And then I'll be really leaning on the biologist to say, hey, what do you think this animal might have been exposed to? Was it a heavy metal? Was it an organic contaminant? Because depending on the toxin, that'll dictate what kind of tissues we need to harvest, how we need to submit it, prepare them for laboratory analysis, and then how they need to be analyzed. So again, toxicology is not something that is done unless you really have a good reason to think that you might have an animal that's poisoned by something. And then molecular biology can be very useful as well. So oftentimes now people go straight to PCR. They'll PCR something and get a sequence, and they'll say, hey, we have it. We have it by PCR. We got the sequence. There's no doubt that this is a cause of death. But remember, all molecular biology does is to give you a sequence. It doesn't tell you whether that sequence is from an organism that's alive, let alone an organism that's causing pathology in the animal. So just because you find a sequence in an organism doesn't mean that's the cause of death. You have to be able to relate that back to what's killing the animal. It's a really good example of how this derailed an investigation is sea-star wasting disease. I don't know if some of you might be familiar with that. There was a big die-off of sea stars in the Pacific Northwest of North America back in 2014, and it killed a bunch of sea stars up and down the western coast of North America. And folks did investigations, and they went in there and they found these sea stars that were wasting away. And they did PCR on these sea stars, and they found viral sequences and they concluded that viruses were the cause of sea star wasting disease. But nobody really went in and made a link between the virus and cellular pathology in the sea stars. And it turned out down the road that the virus has absolutely nothing to do with sea star wasting disease. So this is a danger that going straight to molecular biology can lead you to unless you relate it back to what's happening at the tissue level. Again, re-emphasizing the importance of pathology. There's some other tools that we use for vertebrates, things like fish or higher fish, turtles, birds, mammals, and that's serology. So when you look at animals, when they get exposed to an infectious agent, it will mount an immune response to that infectious agent in a form of two ways. They'll either throw cells at the organism, or they'll throw antibodies, which are special proteins that will mount an immune response against that organism. And you can detect those antibodies using special assays for those proteins. And those antibodies, which are proteins, are very specific to certain organisms. So for example, if you have an animal that's exposed to, we'll say, circles and crosses, that animal will mount antibodies that are specific to circles and then specific to crosses. So you can basically measure the levels of those antibodies and determine whether an animal has been exposed to that particular organism. So for example, if you go to habitat and you're interested in whether crosses are circulating in that animal population, you can take a blood sample from that animal and determine the level of antibodies to crosses. And then you can say, hey, if animals have antibodies to crosses, then crosses must be circulating in habitat. So it's a really nifty way to evaluate whether an infectious agent is circulating in the habitat. It's kind of akin to humans, right? If you take a blood sample from a human, you can tell whether that human has antibodies to the AIDS virus. If that person has antibodies to the AIDS virus, you can be reasonably confident that the AIDS virus is circulating in that population of animals. COVID is another good example. So this is useful for determining whether infectious agents are circulating. It doesn't tell you whether the infectious agents are actually killing animals. All it tells you is, yep, it's in the habitat and you need to be aware of it. And then the last tool that we use, again, for things fish or above, is hematology. And that's where you look at the blood cells of the animal and you can examine the blood cells and you can see whether they look abnormal or not, whether the numbers of blood cells are abnormal. Sometimes you can look for things like parasites that affect blood cells, like malaria. So that can be useful for certain processes. And it's another tool that's an adjunct that we can use to assess the health of wild animals. Obviously, in order to get a blood sample, you have to be able to restrain the animal, and you have to be able to get the blood sample from the animal. So for things like corals, obviously something like that would be useless. Again, just to reemphasize that all of this hinges on getting specimens that are suitable for examination. And this can be a real challenge in wildlife, right? So unlike farm animals where you have cattle in a pen, a cow dies, the farmer sees the cow, calls the veterinarian comes out, get access to the cow right away. That's really easy. In wildlife, getting access to fresh specimens can be really difficult. You have to go out in the habitat, you have to go out in the forest, you have to find the animals that are dead, and you have to find animals that are fresh dead. The more decomposed the animals, the less likely we're going to be able to figure out the cause of death, because decomposition compromises the integrity of the tissues. And interestingly enough, when you look at animals, different groups of animals can sit in the habitat different periods of time. So for example, mammals and birds can afford to sit in the habitat longer before decomposition than, say, things like invertebrates or fish. If a fish dies, a few minutes after it's dead, it's not going to be very useful for necropsy because they decompose really rapidly. Invertebrates, you almost have to get them while they're alive. If you get them after they're dead, they're essentially useless. So again, getting fresh specimens is really, really challenging. And I recognize that, having worked with many biologists, having worked in the field, I know it's difficult to get fresh specimens for necropsy, but you got to make the effort if you're going to try to get an understanding of cause of death, because if that animal is decomposed, if I can pour that carcass out of a plastic bag, I can guarantee you I'm not going to get any sort of useful information for laboratory diagnostics. So I recognize the effort, I respect the effort, I know it's difficult, but it has to be made in order to get specimens suitable for diagnostic investigations. This graph kind of gives you an idea of return on investment of all these different laboratory tests I was telling you about. So on the left axis is the cost. The higher the cost, the taller the axis, the higher the cost. And on the horizontal axis is the amount of information you get for that cost. So pathology, as you can see, is really a low-tech, low-cost approach. All you need is a jar of formalin and some necropsy tools, and you're set. I've taken my necropsy tools in very remote situations where there's no electricity, nothing basically, and I've been able to get some really good biological data from necropsy information. Toxicology and molecular biology, on the other hand, are very high cost. And basically, it's a good contrast. From a microscope slide that costs me about $6 to process, I can give you information as to potential etiology, tissue affected, how that could affect the anatomy of the animal, and how the animal is responding to that insult. With a molecular test, all I get is a sequence. That's it. I have no other information. So, bang for the buck, I would say pathology historically has given me the most useful information. And recognize, I'm not going to oversell all of this stuff. I'm not going to lead you down the primrose path and tell you, hey, do this, and I'm going to guarantee you to get an answer, because there's many times when I can't get an answer. But I can tell you, in most cases, what didn't kill an animal. And that, from a management standpoint, can be just as useful. Because when you think about it, when a biologist is faced with an animal mortality, there's a lot of pressure on that biologist. There's a lot of people saying, well, figure it out. What's going on? I think that's killing, I think this is what's killing the animal. I think that's what's killing the animal. Everybody's going to come out from the woodwork with their little pet hypothesis as to what's killing the animal. And if the biologist can come back to the politicians and say, okay, look, we don't know what killed this animal, but we can tell you for sure it's not an infectious agent. It's not a bacterium. It's not a fungus. It's not a parasite. That can be very valuable from a management standpoint, because then next time you know that you don't need to focus on those aspects. You can focus on the other aspects that could be more probable. So how does this work in corals? Well, to recapitulate, you have the field signalement. You describe what's happening at the morphology level. You arrive, hopefully, at the etiology, and if you don't, you keep turning that crank until you can arrive at a cause of death, and that's what we're trying to do with SCTLD in the Caribbean. Okay, so you got to remember that in animals, disease is not a binary process. It's a continuum. So when you have an animal that's perfectly healthy and you have an animal that died from whatever, things change over time with that animal from here to there. And so it does in corals. So one of the big fallacies people have in corals is they'll see a gross lesion and they'll infer causation based on what they see grossly in that animal. And that's analogous to me being able to say, hey, this bird died from an infectious agent by just looking at the bird that's dead. There's no way I could tell you that this bird was dead from an infectious agent without doing laboratory diagnostics. And recognize that even though a lesion looks different over time, that lesion could be caused by the exact same thing. You're just looking at a different stage of the disease over time. So it's really difficult when you're going out on a dive and looking at a coral and looking at a lesion to say at what point in this disease is this animal occurring? You don't really know other than what you're looking at grossly, and there's just no way that you can infer the cause of that coral lesion without doing laboratory diagnostics. The only exception to that would be predation. So if you see fish bites, clearly fish bites is sort of trauma on a coral. You can diagnose fish bites if you know the habitat and you know the fish. Crown of thorns starfish, that's another really good example. If you see a starfish and you see tissue loss in that coral, clearly that's predation in the coral. Okay, so there are things you can diagnose, but those are very few and far between. Most of the time, you're going to have to take samples of the corals and do laboratory analyses. And so that's what we're doing that's what we did in Florida with stony tissue coral loss disease. We took samples of corals, we photographed them, we photographed the lesions to characterize the gross pathology, but critically, we then took tissues in fixative, we decalcified them, and then we characterized the pathology at the cellular level. So corals, unlike sea turtles or fish that are much more complex with organs, livers and hearts and gills and kidneys and muscles and all these wonderful organs that we can look at, corals are a lot more basic. It's basically a polyp sitting on a calcium carbonate matrix. So we don't really have very much to work with other than a polyp architecture. And if you take that polyp architecture and you dive down into it, basically that coral polyp, if you take that blue rectangle here, so you have these polyps here with their stomachs that are interconnected, and they have gastrovascular canals, which are basically their stomachs that are connected between the polyps. If you take that little blue rectangle and you look at that at the cellular level, there's basically only 3 tissue layers that we know of. There's an epidermis with the stinging cells. There's a connective tissue layer. There's a gastrodermis that contains a symbiotic algae. There's a stomach or gastrovascular canal. There's another layer of cells. And then there's another connective tissue layer. And then there's a calicodermis that secretes the coral skeleton. So basically, when we do histopathology in corals, that's what we're looking at. We're looking at those tissue layers, and we're looking at abnormalities in those tissue layers and trying to characterize that at the cellular level. So what's happening with SCTLD? Well, based on what we've seen on histopathology, what we're seeing is we're seeing damage in the zooxanthellae and in the host cells. Now, because these are symbiotic organisms, we know that the health of the symbiotic algae and the health of the host cell that houses that symbiotic algae are very connected, right? The health of one is totally connected with the health of the other. And what we're seeing is that we're seeing a breakdown in the health of these symbiotic algae and the host cell, principally at the basal tissue level. So what's happening is that there's pathology in the zooxanthellae that we're seeing at the light microscope level. There's pathology in the host cells at the light microscope level. And what that's leading to is death of that basal tissue layer. And then that seems to spread to the upper tissue layers, and eventually the tissue slough off, and that leads to the clinical signs that we're seeing in the field, which is the stony coral tissue loss disease. So remember that gross lesion is a very crude indicator of what's happening. Basically, it's just death. It's tissue death. And there's many things that can cause tissue death. We haven't seen anything at the microscopic level like bacteria or fungi or parasites that could lead to that kind of lesion. All we're seeing is pathology of zooxanthellae and pathology of host cells. So it appears to be starting with the zooxanthellae at the microscopic level. When you look at what looks to be early lesions, the pathology seems to be starting with the zooxanthellae, but we're really not sure about that because remember, we've only sampled corals at one point in time. We haven't had the luxury of sampling an animal over time to see how this disease progresses. So it's analogous to you've got a person on a necropsy table with a shrunken liver. Well, you don't know how that person got a shrunken liver. It could have gotten a shrunken liver because it had hepatitis. They could have had a shrunken liver because the guy was an alcoholic. You don't know until you look at that person over time to see the development of that lesion in the liver over time. You're only seeing that in one snapshot. So that's one of the crucial things we need to do is we need to be sampling animals over time to see how this disease evolves. Okay. We also know that apparently normal corals have lesions. So we've gotten samples from corals that grossly look, and that, you look at them in the field and, hey, yeah, that looks like a normal coral. But when you look at them histologically, there's definitely pathology happening in the zooxanthellae in these corals. So, there's something going on before these corals lose that tissue. And because we're not seeing anything at the light microscopy level that could explain this in terms of infectious agents, we're not seeing bacteria, we're not seeing parasites, we're not seeing fungi, then we have to think that the only other possible causes is either something that's not visible at the light microscope level, maybe a virus, maybe a toxin, maybe a toxicant. We know that these zooxanthellae are dinoflagellates. Well, dinoflagellates can make all sorts of toxins. We know that from harmful algal blooms. Maybe there's something going on metabolically where the dinoflagellate is making a toxin that then is killing the host cell. So one of the critical questions to answer with stony coral tissue loss disease is there's a breakdown in the symbiosis between the zoox and the host cells. The key question is it the zoox that's going down first, leading to death of the host cell? Or is somehow the host cell doing something that's inhospitable to the zooxanthellae, leading to the death of the zooxanthellae? That's a really critical question to answer. We also have clues that it appears, at least at the histology level, that not all species are equally susceptible. And biologists have known this from field observations as well. This disease doesn't seem to be affecting all coral species equally. For example, Acropora, which are typically the ones that are most often affected by tissue loss diseases in the Caribbean, they seem to be doing fine, which is really weird. So one of the next steps we're doing as we speak is we're doing electron microscopy. So electron microscopy is a way to look at the cellular details in even more detail than what you can see in light microscopy. So on light microscopy, this is the kind of detail you can see. You can see the cell, and you can see the nucleus, basically. With electron microscopy, you can not only see the cell and nucleus, but you can see all these little different organelles in the cells because you have much higher resolution. And I might add, you can see things like viruses that you can't see at the light microscope level. So that's one of the things we're doing right now to at least try to rule out maybe the potential that viruses might be involved in stony coral tissue loss disease. So some of the questions, what kind of organelles are being affected? Is there evidence that a virus might be involved that's not visible on light microscopy, but we could see perhaps on electron microscopy? And if yes, the two above, then what cells or organelles are this virus infecting? So if it is an infectious agent, then that's great, because then at least we can hopefully confirm, we can develop a PCR assay for this agent. We can start tracing it in the environment. We can localize it to tissues. We can develop a diagnostic test and trace the ecology of this infectious agent. At least that gives us something to hang our hat on. If it's something non-infectious, like a toxicosis, that's going to be a lot more difficult, because then it's going to take a lot more work to try to determine what would be that non-infectious cause. What kind of toxin would that be? What's producing the toxin? How is this toxin killing the corals? That would be, a very difficult thing to answer. And so that's what we're going to do in Puerto Rico and the Virgin Islands. We're going to go and we're going to collect samples in fixative. We're going to look at these tissues under the microscope to see if the lesions we're seeing in Puerto Rico and the Virgin Islands are the same thing that we're seeing in Florida. Maybe it is, maybe it's not. We're going to take samples for electron microscopy and then basically try to replicate what we did in Florida to see if we're looking at the same disease in Puerto Rico and the Virgin Islands that we're seeing in Florida. And with that, I think that's all I have, and I'll open it to questions.
Great. That was really clear and really educative. Like I really appreciate the information. And so let's go ahead with any questions from participants. I saw someone raise their hand earlier, but didn't ask the question. Is that person still? Was Ernesto?
No, it wasn't
Hello?
me. I just raised my hand. It was somebody else before, yeah.
Okay, but do you have a question, Ernesto?
I just want to compliment a little bit look what Thierry said about what we're planning to do here in Puerto Rico. One of the main aspects, I think, is that we're going to follow some of the colonies over time, collecting samples while, first, the colonies, then when it is diseased, and then afterwards to see whether, what's the progression of the damage to the tissues. And thank you, Thierry, for a wonderful talk. Very
yeah,
illustrative.
you're welcome.
So Terry, when you talk about looking at the tissue samples over time, is that what is referred to as pathogenesis?
Yeah, kind of. Pathogenesis is more, yes, what is the progression of events that lead to a lesion? So, when you see a cell that's dead, there's a whole series of events that occur to lead to the death of that cell. And that is disease pathogenesis. And one of the reasons we want to sample things over time is that that example I talked about earlier about the guy who's dead with a liver lesion on the necropsy table. You don't know how that animal or that person died from shrunken liver. He's just got a shrunken liver. And so it would have been really useful for that person if you'd been able to take blood samples from that person to see the evolution of the liver disease, to see that, oh yeah, there was a hepatitis virus that came in and caused the liver lesions. Because sometimes what happens is that when you look at a lesion at one point in time, it's like looking at a forest fire, and the forest fire has gone by, and you're looking at the dead forest, and really what you need to do is you need to go to the edge where the forest is burning. And so maybe, looking at different stages of that disease, you'll be able to catch something in action, whereas if you only look at it at one point in time, you may have missed the boat. Sometimes an infectious agent comes in, causes the mortality, and then leaves, or disappears, and then you're left with the lesion. So this is the critical part that Ernesto was talking about. It's that, we did this in Hawaii where we sampled corals over time, and we got a lot more information about what was going on. We saw things coming in, we saw things leaving, and it was just a much more dynamic process than just taking a photograph at one point in time.
Yeah. Excellent, thanks. So we got a question from Dr. Rebecca Gibble. Rebecca, do you want to make the questions using your microphone, or you want me to read it from the chat? Okay, I'll take.
Hi. Perfect. Yes. I heard in the most recent notes that you're trying to replicate in Puerto Rico and the Virgin Islands what was already done in terms of electron microscopy sampling in Florida. So am I mistaken, Dr. Pitlick, or did you already do EM on Florida SCTLD samples?
I'm in the process of writing the electron microscopy report as I'm talking to you guys right now. So I'm hoping the report's going to come out. It has to come out by mid-June, because my neck is on the line to get the report out. So yes, we have done EM, but I don't want to divulge the information until I talk to the Florida people first, just out of respect for there. But it's going to be public information, so it'll come out pretty soon in any case.
Okay, great. Well, we'll eagerly await that. Thank you.
Great. Okay, so we got a question from Joe Townsend from the USVI. Will you Joe, you want to make the question yourself?
I'm on the road, so yeah, actually, it would probably be better if someone else could just read.
Okay.
Sorry.
Thank you, Joe. Sorry about that. So Joe asks, will you be sampling anywhere in Puerto Rico where disease has not been reported at all?
I think that would be really important, yes, because one of the big questions we have is, we've had very few corals in Florida that do not have lesions. So, whatever is killing these corals in Florida seems to be, clearly it's widespread because we already know that corals are dying all over the place in Florida. And so I'm really curious to see in Puerto Rico, can we find corals that don't have lesions in Puerto Rico? So yes, absolutely, we want to do that.
Yeah, definitely, and specifically on species that are highly susceptible, like Dendrogyra, Meandrina.
Absolutely.
Those
Absolutely.
would be really nice. So we got a next question from Judy Lang. Judy, you want to open your microphone?
Miguel, I think she said go ahead and read it.
If you don't find
Okay.
smoking gun
Oh, yeah.
particles in the air,
Okay.
how
Sorry,
would
I
you
didn't.
start to try to distinguish between an external toxicant like a pesticide and some internal toxin coming from the symbiote? You mentioned that's very difficult.
That's going to be extremely difficult. It's going to take. I can tell you that we discovered a marine toxin that was, that pufferfish photograph I gave you earlier, that was caused by a marine toxin. It took us 5 years to figure that out. Discovering new toxins, especially if it's a natural product, is a very labor-intensive process. It's a lot of analytical chemistry and chemical synthesis. So yeah, I mean, if that's where the EM points us to, then we're going to have to go down that road. There's going to be no question about it. If it's a pesticide, really that depends on, we need to find a candidate. What pesticides are being used where? Can we find epidemiological patterns where pesticide use is overlapping or herbicide use is overlapping with the stony coral, tissue loss disease? And again, that takes a lot of field work, a lot of mapping, a lot of shoe leather work, basically, to sort that out. Toxicoses are really difficult to sort out.
Right. So Joe comments that in the field they have an observation where they're looking at a rise in the frequency of subacute bleaching lesions with little to no mortality at all. And this is especially after 6 to 8 months of the first lesions being observed. So he mentions that it will be interesting to compare those lesions to lesions with actual rapid tissue loss, especially over time. Okay, yeah, so yeah, in Puerto Rico, we're looking at similar things. I don't know if after tissue loss lesions, but we're looking at definitely some coral, especially from Orbicella, that we're looking at bleaching that is, adjacent to the tissue loss line of the disease. And sometimes the bleached tissue can stay for a couple of months without the mortality progressing significantly. So definitely a lot of variability in the gross morphology of the diseases that we're looking at doing these outbreaks. Thanks, Joe, for the observation. So Ernesto Well asks, have anybody analyzed water samples for toxins from areas with high SCTLD mortality? Looking at toxins in the water column. Oh, we just lost Dr. Work. Maybe he had an issue with the connection. And Ernesto, I heard from a meeting with Florida colleagues that actually some people were looking into harmful algal blooms and maybe some toxins released during those disturbances. We got Dr. Work back.
Sorry, my internet connection went out. I apologize.
Oh, no worries, no worries. Yeah, I was reading Ernesto's question about if anybody have analyzed water samples for toxins in areas with SCTLD mortality.
Well, the problem with that is what toxin are you going to look for? You know, we know that there are dinoflagellate toxins that are produced in the Caribbean. The question is, you know, there's many more toxins that these things make that we don't even know about. And so, yeah, I don't know. I don't know of anybody who's analyzed toxins in relation to SCTLD. I'm not aware of anything like that.
Yeah,
I was mentioning, Dr. Work, that I believe some. Oh, sorry. Go ahead, please, Ernesto.
No, that's okay. I was just thinking about any, general screening of water samples. I mean, I know people is collecting water samples, but they're looking for basically pathogens and bacteria and stuff like that. But I don't think that anybody's thinking about what you mentioned, any kind of toxicants or toxins. And that's what I asked the question.
Yeah, and even if you see it in the water column, I suspect that if it is a toxicosis, because it would be something locally produced inside the coral that's killing the cells around the dinoflagellates. So the dinoflagellate would produce some sort of toxin that then kills the host cell. So I don't think you'd even see it in the water column. You'd have to actually have to extract zooxanthellae from the coral and extract, and do the analyses on these zooxanthellae to find the chemical product that could then might be responsible for that. Cyanobacterial toxins, I mean, Judith is mentioning that. Yes, but there's no evidence of cyanobacteria in these corals. I mean, black band is a really good example of a cyanobacterial disease that definitely kills corals. And it would make total sense there that cyanobacterial toxins might be involved, but not in this case.
So, Thierry, I mean, you sort of imply that no basic bacterial virus pathogens.
I'm not saying virus.
Infectious.
I'm not saying
Okay,
virus.
yes, no virus.
The things we have pretty definitively ruled out are bacteria, fungi, and parasites, at least based on what we see on light microscopy.
So, to explain the transmissibility and the infections, then we have to think about that possibility of viruses.
Well, yes, but remember, transmissibility is a really tricky thing because, for example, C-star wasting disease, people thought that was transmissible, but it is absolutely not transmissible. In fact, you can put a 6 C-star, or you can put a C-stars, for example, will drop off their arms, and that's part of their natural defenses. And when they do that, they elicit hormones that go out in the water and promote other C-stars to drop their arms. So you can look at that and you go, that's infectious, but that's a purely hormonal mediated process. Plants do that. Plants will slough their leaves based on hormonal signals and hormonal signaling. So I would be, just because something looks transmissible in the field, I would be wary of just concluding that it's infectious until you actually are able to identify the infectious agent associated with that lesion.
Yeah. Correct. So we did some experiences also with yellow band disease, and we never actually found with this was John Brune and myself, and we couldn't get any transmissibility. However, the disease looked like if it was infectious because it show up in large geographic areas everywhere. But that could have been triggered by any changes in environmental conditions, or for any of the mutualistic bacteria that are living within the coral normally, and then they become virulent for some reason. Yeah.
Right.
Yeah.
Okay,
Thank you.
awesome. Well, thank you guys for the discussion.